Today's topic is going to be about the cell membrane and how the cell membrane controls things that go in and out of it and the processes that occur because of that. So here the first thing we're going to look at is the actual structure of the cell membrane, and sometimes it's called the phospholipid bilayer. And the main component here, which may look familiar to you, these little circles. This is a bilayer because look at, there's a circle and two lines that come off of each circle. That circle represents the phosphate group of a phospholipid, and the two lines represent each of the fatty acids that are also attached to a glycerol molecule. And the glycerol molecule is not drawn, it is assumed to be there, but this is just a very basic diagram, and when you get basic things, they just kind of only are giving you the important stuff there, so they highlight the phosphate group and the fatty acids. So there's a layer on the bottom, there is a layer on the top. Other characteristics you have to note the fact that why are the phosphate groups? Think what's below this, and it doesn't necessarily in this picture give you the perspective that they're doing, but for example, like this might be below the membrane, might be outside of the cell. Above the membrane might be inside the cell. Or vice versa. I'm assuming because of the way that these other parts are, that this is most likely actually reversed, that this is going to be outside the cell and this would be inside the cell. The basic component of what's inside the cell, which is cytoplasm, is going to be water-based. Outside of the cell, also water-based. So the chemical properties of a phospholipid is the fact that the phosphate group, that is hydrophilic. It has a charge, so therefore it interacts with water. The fatty acid tails are hydrophobic. They are nonpolar, so they don't interact with water. So look at how the phospholipid arranges itself. The hydrophilic are on the outside of each side, so that therefore they're in touch with the water, and the inside of the phospholipid bilayer is particularly the hydrophobic, or is not going to touch water. Other components within the cell are going to be proteins. All these blue structures here are proteins, and there's two different types of proteins. They call them either integral membrane proteins or peripheral membrane proteins. Integral just means that it goes through the entire section of the phospholipid bilayer, and here's one that was cut in half, so you can kind of see it's somewhat of a tube. That's going to play an important role in one of the processes of the phospholipid bilayer. Peripheral membrane doesn't go through the entire membrane, it just kind of hangs out on one side or the other. So these ones in the back, you really can't tell if they're technically going through or not, but that's the major difference between peripheral. Think of periphery like on the outside, like your peripheral vision, the vision that you can see outside. Perimeter, outside. Integral means to go between. So this is an integral protein. There are other components that the more biology you learn, the greater detail you go into, but for right now, the two most important aspects of this diagram are going to be the phospholipids, which once again are here, and the proteins. All right? So that covers the basic structure. The next thing that we'll talk about is going to be the different processes that occur across the membrane. So I have a few animations, and I'm not going to press play. I'm actually just going to fast forward them and somewhat describe them as they're going through. Here's the first one, and the first process is called diffusion. And this fits under a category of passive transport. Passive just means that there is no energy required. It's somewhat an automatic process. It will happen with or without energy available. And diffusion is the process by which molecules are going to move from an area of high concentration to an area of low concentration. So this animation is going to show that. And there's two different ways that it can be described. This is just diffusion within a space, and then there's diffusion across the membrane. I particularly think that diffusion across the membrane is going to be more important for this chapter, but also realizing that diffusion happens even without a membrane present. So here's a beaker with water, and inside of this are these solutes. So recall that solutes were things that get dissolved in water. In this case, we have water, which you can't really see the molecular composition of it. You just kind of recognize that there's water in there. And then they have these yellow solutes. So we're going to dissolve the solutes into the water, and they're going to show you how they do that. And these molecules are constantly moving. So here is somewhat a state of what's called equilibrium. The molecules are moving all around. There's no real space that is more concentrated than the other. But how did they get to that spot? And this animation shows it. So here's a lump of sugar, which is a very highly concentrated area of solute molecules. Here's our water. So what will happen automatically, once the lump of sugar is dropped into the water, then what will happen is these individual molecules, they're not chemically combined, they're all just physically interacting with each other, so they can be separated. So what they'll do is they will separate. They're now starting to go from where there was a high concentration, there is little to no concentration of solute out in this area. So they start to travel to where there is a less amount. So there it says high to low concentration. That's how I know it's passive transport. Going from high to low does not require energy. Just like if you had a hill and you dropped a ball at the high point, it will go to the low point automatically. So this will go until it reaches equilibrium, and that's the state where everything's at somewhat an equal concentration. And it just doesn't stop there and say, all right, we're done. It will continue to move around forever. They just stay, they tend to have, as one molecule leaves, another one kind of takes its place in that same area. So you never really go back into an area where there's going to be little of that solute. Okay? That was just an animation to show how it happens without a membrane. This little animation shows how it happens with a membrane. So here's our phospholipid. Look at this diagram. Even just really highlights the simple stuff. The phospholipids, phosphate group, fatty acid tails. Here is their version of an integral protein. There's a lot of other things going on here. I really want to highlight just what is known as simple diffusion. That means it's going through the membrane. So I'm going to press play and see if I can speed this process up. So notice, this is the molecule that we're kind of looking at. These molecules that are going right through the membrane. They even highlight here, hydrophobic tails. This other molecule, there's some molecules in here like glucose. Glucose cannot go through this membrane via simple diffusion. That's what we're focused on right now. It is too large to get through. But look at this molecule. This is probably it actually identifies it as oxygen, something very small. Here's CO2. Can that get through? According to this diagram, it's small, and it gets through via the squeezing between these phospholipids. There are chemical properties that take place because of this. In order to actually get across the membrane, you have to be small and you have to be nonpolar. That's the two requirements to go through what is called simple diffusion. So oxygen only has 2 atoms, O2. CO2 has 3 atoms. They're small enough to squeeze through these phospholipid bilayers. They have to be nonpolar. Polar molecules cannot get through. The polarity is kind of rejected by the interior of this membrane, so therefore they kind of are repelled by it. So something like water. Water has 3 atoms to it, which is small, but water is polar, so therefore water can't get through. This diagram doesn't show the best in terms of concentration. It mentions it just in kind of terms. I don't know if you necessarily can see the molecules, but if the higher concentration is here for oxygen and the lower concentration is down here, then the movement is going to be from above the membrane to below the membrane. CO2, in that one example that I kind of saw as we were going through this, I don't know if I can remember. Yeah, I can. CO2 was going from below the membrane and was going out of the membrane. So that must be assumed then that there's a high concentration of CO2 down here and a low concentration up here. So this will continue, and just like when you get to the end of a diffusion in a non-membrane example, molecules will still move, as will the case when this gets to equilibrium. Once oxygen diffuses enough and there's an equal amount of oxygen on both sides of this membrane, oxygen will somewhat go back and forth equally. It doesn't mean that now there's a barrier that says no more across. They will move back and forth in equal concentration so that above the membrane and below the membrane would be somewhat called at an equilibrium. All right. The next one is a little bit trickier, I will say. This is what's osmosis. Osmosis is the diffusion. So as soon as you see the term diffusion, that really means you're going from high to low with no energy. Osmosis is a particular movement where water is going to go across the membrane. So here's an example. It's a non-biological membrane, but we're going to first start with this, and then eventually we'll talk about what it looks like across an actual phospholipid bilayer. So here we have our somewhat selectively permeable membrane, and on either side of the molecule, I believe that these are recognized as okay, I'm just trying to get ahead of. So this was showing, I guess, a little bit of simple diffusion. It says in the thing, diffusion is the net movement that allows, this process allows small molecules such as oxygen and carbon dioxide to cross the plasma membrane. So here, polar molecules, they kind of change colors on us and say that such sugars and proteins, they can't go across because they're polar. Now they focus it on water. So this animation kind of does a lot of skipping around, but try and hopefully follow my instruction. Here we have water. Water is polar, so therefore should not be able to get across a membrane by simple diffusion. Instead, they are going to need special proteins. And this diagram or animation doesn't show that. In an actual phospholipid, I can show you where water molecules would get across. So in this animation, you have to have the imagination that water is traveling across this. It's allowed to get across as long as there are potential holes built for polar molecules to get across them. So what we'll do is water is showing it's going back and forth. All right, both sides are getting there. Now they're going to bring into the mix this molecule, which is our solute example. All right, water then can travel back and forth. But what will happen is both sides of this membrane are no longer in equilibrium. We're going to look at both sides of this membrane in terms of its concentration of what are known as the solvent concentration and the solute concentration. There are things that I obviously would love to change about this animation because they don't put any urea over here on the left-hand side. So that somewhat means that the concentration is 0 for the solute and 100% for solvent. Over here, there is definitely not 100% water. There is somewhat a concentration of solute particles, which in this case were the urea molecule. So I somewhat make up numbers in class or to show this process, but if I make them up right now, I'm going to have to assume that we can't make this pure. This is somewhat, it makes it much more difficult for you to grasp if you think about this as 100% pure water. So say that I said that this was, 90% water and 10% solute that technically you can't see. Over here, because there is a lot of solute, I'm going to make the solute number like 50%. That would then make the water concentration 50%. So what's going to happen is water wants to get into equilibrium. So if there's 90% water here and 50% water here, water always travels to where there is less of it. So 90% left, 50% right, water is going to travel to this side of the membrane. So if I fast forward this, what will happen? There it goes. So the arrow shows that's the direction water is going to travel. And the reason why it's traveling there is because there's less water concentration here. And what it should do is it should go until it technically balances. So if you kind of saw the number 90 here and 50 here, think of a balance. I can't do that. I guess that would be 70. So until it reached 70% water on both sides of the membrane, it hasn't reached equilibrium. Water will continue to travel to the right. And because of that, the volume is rising on this side because all the water is traveling to the right-hand side. So that will occur. Now they do an example where they switch it around. Look at what happens. The left-hand side has the same amount of solute concentration as the right-hand side does. So therefore, our numbers are going to be identical. So I can kind of say this is 50% solute, 50% water. 50% solute, 50% water. And if I continue, this is just going to show that water is going to travel in both directions. It should basically travel in both directions because it's at equilibrium. There's an equal concentration on both sides of this membrane. So there's a term that they describe here, and that's the word isotonic. And isotonic just means that there's an equal amount of water on both sides of the membrane. Hypertonic and hypotonic are terms to describe when you have more or less water. So hypertonic here, hyper is going to mean more, but that means there's more solute, and more solute means less water. The other one was hypotonic, and hypotonic meant that there was less solute and more water. So that would have been this side originally.